So for this week's lab, you'll first need to download the popG program onto your computer. It's free and a pretty quick download, and if you're working along on your own while watching this video, you should be able to run it in a separate window. Now if you run into any problems, make a note of them and bring them up when we have our live Zoom meeting for lab. So once you have a basic understanding of the microevolutionary forces, this app is a great way of reinforcing and self-assessing your knowledge. This is the whole point of my having you do this lab exercise. But if you haven't gotten yourself to the point where at least natural selection and random genetic drift makes sense to you, you should wait until you do have that basic grasp before you start with this lab activity. I don't think that you'll learn much from just going through the motions. You need to understand why we're manipulating the simulation settings the way that we do and what we're intending to show from each of these simulations. Okay, so presuming now that you're actually ready to reinforce a basic understanding of random genetic drift, I'll quickly go over the popG app. There are only two windows. The output, that's the one you're seeing here, and the settings window, which you get to by looking under the Run menu. Let's start by putting up a result to interpret. Start with the default settings, but change the number of populations evolving simultaneously, this one right here, to 1 rather than 10. Then press OK. When I do this, my popG settings window disappears, so I'm bringing it back by selecting New Run. It will have kept the same settings that I used in the previous run. See that the number of populations is still set at 1. So these results in the output window correspond to the settings in this window. Now looking at the output graph, mine shows a squiggly line starting at generation 0 with p equal to 0.5. That's because in the settings, the initial frequency of allele A is 0.5. If you wanted to change this to any value between 0 and 1, you could do this from the settings window. For now, we'll just leave it at 0.5. The thin black squiggly line gives the history of change of allele frequency of big A over time, over 100 generations, which is another default setting that you can change in the settings window if you want. The thicker light blue non-squiggly line shows the expectation of a population experiencing no genetic drift. Now, this would correspond to a population of size n equal to what? So if you can't answer this, it probably means that you haven't gotten there in lecture yet, and you might want to try this again after completing the lecture content first. But if you said that you have no drift only if n is infinitely large, n is equal to infinity, that's great. That's the right answer. And yeah, our population size is 100, which is definitely smaller than infinity, and so there will be genetic drift, and that is actually the cause of the change in allele frequency we're seeing in this graph. We know it's not natural selection because the fitness values for all three genotypes is set to the same value, 1.0. We know that mutation and gene flow are not factors here because mutation and migration are both set to 0. And in the absence of natural selection, mutation and gene flow, the only thing that could possibly change gene frequency is random genetic drift. And we know there will be drift because the population size is 100. Now with this simulation program, you have control over the microevolutionary forces. You can manipulate population size, fitnesses, mutation and migration rates. You can adjust where you want P to start at generation 0. You can change the number of generations shown on the horizontal axis. And you could also have the output show multiple simulations all starting with the same exact settings. Now, if I press OK again on the settings window, I'll get another run with the same settings. You can repeat a run with the same settings this way by using new run that repeats the settings from the previous run, or you can also use restart under the run menu. Now you see here that when I repeat a run with these settings where there's only genetic drift affecting allele frequency, I'm not getting the same result each time. In this run, after just over 60 generations, the allele frequency of big A went down to 0. But this also means that the frequency of little a went up to 1.0. At around generation 62, I think, the population became fixed for little a. Every individual of the 100 was little a homozygous. Note that one thing this graph is most definitely not telling us is that the population died out, because it didn't. It's there throughout the entire graph with a population size of n equal to 100 individuals. The difference we're seeing between generation 100 and generation 0 is that at 0 we had 50% big A and 50% little a, whereas at generation 100 we have 100% little a and 0% big A. When I run this again, I end up with a result showing that even after 100 generations, the population hasn't gone either to the top or the bottom. Looks like there's still around 70% big A and 30% little a. Now, under the Run menu, you have the option of continuing for another 100 generations. As you can see, this graph picks up at the same allele frequency, 70%, where the first one left off, and the generations go from 100 to 200. And after 200 generations, I still have both big A and little a segregating the population. But when I keep going for another 100 generations, it looks like at around 240 generations, the population has gone completely fixed for big A. Every one of the 100 individuals in the population is a big A homozygote, and the population is basically stuck at p equal to 1.0. Because if everybody is big A, then all of their offspring also have to be big A, and there can never be any little a genes again unless they get introduced by mutation or migration, but we'll get to that later. Now, if you go back to the default settings and hit OK, you'll get a plot with 10 results, all starting with the same settings, plotted on the same graph. Okay, now copy this graph into your lab notes. You can do this with a screen capture or something. This graph gives you a feel for how much fluctuation you can expect to see due to random genetic drift for a population of size n equal to 100. Now, we're going to compare this graph with what we get if we increase the population size fivefold to n is equal to 500 and decrease the population fivefold to n equal to 20. Do both of these runs and incorporate the images into your lab notes. Make sure to label what it is you're showing with your image. Here, the most important bit is what the population size is for each of the simulations, whose results you're recording with your images. The biggest common mistake that students make with their lab notes is including some kind of picture with no information about what the picture is supposed to be showing. Don't make the mistake of doing this. In the first item of the accompanying lab worksheet, paste in your images, all three of them, as part of your response to these questions. How did increasing or decreasing the population size affect the magnitude of the zigs and zags in the histories of allelic changes due to random genetic drift? How did increasing or decreasing population size affect the number of populations out of the 10 that went to fixation after 100 generations? And by fixation, I mean either fixation of big A at the top or fixation of little a at the bottom. The next question in the same item doesn't need an image, but you will need to run the drift-only simulations with n is equal to 20, n is equal to 100, and n is equal to 500 again in order to answer this question. How many generations did it take for all 10 of the populations to become fixed for either big A or little a? Now you can use continue from the run menu as we did before to extend an existing run, or you might need to increase the number of generations shown in the output, especially for where n is equal to 500. Now, based on the results you've reported so far, you should be fully convinced that genetic drift operates more weakly in larger populations. But at the same time, you also know that even for larger populations, the ultimate outcome of drift is the same. fixation of one allele or the other. It just takes longer. All good so far? Awesome. The next thing we'll do is incorporate some weak natural selection into the simulation. Leave the population size at n equal to 500, but change the fitnesses to favor the big A allele, just a little bit. Leave the fitness of big A at its default setting of 1.0, but change the fitness of the heterozygote to 0.99 and the fitness of the little a homozygote to 0.98. This means that the success rate of the big A, the heterozygote, is 1% less than that of the big A homozygote, and the fitness, that is the rate of survival and reproduction, for the little a homozygote is another 1% lower, 98% of the fitness of the big A homozygote. You gotta admit, this is pretty weak selection, right? Oh yeah, and change the number of generations to be reported on the horizontal axis to 750. Now hit the OK button. This time the blue line goes to the top, as do all the squiggly lines. Basically, with the fitnesses set this way, we can expect natural selection to increase the gene frequency to 1.0. And this is exactly what we do see for all 10 of our populations. Does this make sense to you? What I'm seeing is that it may take 500 generations or more, but natural selection is still fixing big A and eliminating little a every time. Now, what happens if the population size is smaller? Let's try the same thing again, except with n equal to 100. Now note that the blue line doesn't change. The expectation based on natural selection has the population becoming fixed for big A, the same as we had before. And yet, for two of these populations, you got exactly the opposite result. Little a got fixed, big A got eliminated. Once you hit the bottom, from that point onward, every individual in the population is homozygous for little a and is less fit on average than the heterozygotes and the homozygotes that were there in the population, even at generation 0. How could this happen? Incorporate these two last graphs into your lab notes and into your worksheet. Make sure that you choose an iteration with n is equal to 100 where you actually get at least one going to the bottom. Okay, it's not going to happen every time. Now, why is it that 10 out of 10 populations went in the expected direction of natural selection when n is equal to 500, but only 8 out of the 10 went to fixation of big A when the population size was dropped to n equal to 100? Answer this on the worksheet. Now you don't have to do this for the worksheet, but do this anyways, on your own. What would you expect if you were to run these same conditions, but for a population size of n equal to 20? Or n equal to 2,500? Why? And did your predictions pan out when you actually ran with these numbers? Okay, let's do a little freelancing. How would you set things if you wanted to simulate a situation in which both homozygotes have lower fitness compared with a heterozygote? Go ahead and do this and use it to demonstrate that this situation where the heterozygotes have the highest fitness results in drift taking longer to result in fixation of all populations. Can you do this? Okay, now let's go back to the default settings. Use this button to restore default. But now set the number of generations to 1,000. Hit OK. With population size equal to 100, most or all of your populations will have gone to fixation at either the top or the bottom well before generation 1,000. Now, let's change the migration rate from 0 to 0.005. That's 1/2 of 1%. This means that on average, Each of the 10 populations receives a migrant from one of the other populations every other generation. How does this affect the result? Grab an image of the resulting graph and write an explanation of what's going on for the third item in your lab worksheet for today. How many of your populations were fixed for big A after 1,000 generations? How many were fixed for little a? And why is there such a great difference between this and our previous result in which there was no migration, where all the populations went fixed for either one or the other allele? Give me a clear explanation. Why does migration allow for the population to reach the top or the bottom and then later go back towards the middle, while going back towards the middle is not possible if there's no migration? Feel free to explore further with this popG simulation program. Remember that the point of having simulation programs like this is so that you can learn by playing scientist. Hypothesize and make a prediction. Test your hypothesis by running the experiment and seeing if your prediction was right. If you weren't right, can you modify your hypothesis to get a better picture of how things really are? By doing this, you should get to the point where, at least for manipulations of the settings for drift and selection, that is population size and fitnesses, you can predict relatively accurately how the populations will evolve. Of course, in the case of drift, you won't be able to predict a specific direction of change for one population, but you should be able to address the amount of random noise around the expectation for natural selection, which is the thick blue line. That's it for today's lab. I'm hoping that you'll come to lab with questions that we'll be able to talk about during our live Zoom meeting.